from Sejong University, and she awarded a PhD in science from Catholic University. Her research interests are induced pluripotent stem cell differentiation and cell therapy. This topic is induced pluripotent stem cell derived minimal injectable unit spheres alleviates cartilage degradation and osteoarthritis. Let's welcome with big hand.
Thank you for the kind introduction, and I would like to personally thank the committee for giving me and my team to present the work that we have been doing for so many years. And today I'll be talking about the developmental process of neuron, which are iPSC-derived minimal injectable unit chondrocytes for osteoarthritis treatment. So I want to first talk about the cartilage tissue. So the cartilage can be found in most of the joints in our body and other organs as well. And the cartilage functions by supporting the soft tissue and provide a framework to the body. And it also responds to mechanical stress and protect the joints by lubrication. So, and specifically, the articular cartilage in the knee joint is consisted of elastic white hyaline cartilage tissue. And as many of you may know, that the articular cartilage covers the end of bones to protect from friction. And as you can see here, the articular cartilage is mostly composed of ECM proteins, and only the chondrocytes are embedded within them in a structure called the lacuna. So this characteristics make the cartilage tissue avascular and a lymphatic characteristics, and this makes the cartilage almost impossible to naturally heal, because when the defect is made, stem cells and other nutrients that has to go to the defected area cannot reach directly when the defect is made. So when a cartilage defect is made, first inflammatory factors are secreted by the chondrocytes within the defect. And these factors induce the differentiation of the chondrocytes and induce the shifting of the ECM component, which makes the cartilage a more fibrotic and calcified tissue. And eventually this leads to bone erosion and spur formation, which narrows the joint space and occurs joint pain. And this is kind of the reason why delaying the cartilage degeneration and also to regenerate the defective cartilage is important, because we have to stop this process. So traditionally, surgical procedures such as microfracture and graft transplantation was performed to treat the defective cartilage, and also cell sources such as the primary chondrocytes and adult stem cells such as mesenchymal stem cells were used for treatment. However, after the discovery of pluripotent stem cells, including ESCs and iPSCs, in vitro chondrogenesis and also the cartilage development are being studied using these cells, and our group as well has been generating chondrogenic tissues using the iPSC. So the articular cartilage is generated through a developmental process called the endochondral ossification. This is also the process that generates bone. And in the early stage of this process, a hyaline cartilage mass is generated through a process called mesenchymal cell condensation. And using pluripotent stem cells, the basic flow of generating chondrogenic tissue for performing in vitro chondrogenesis is to make mesenchymal-like cells from the iPSCs and condense them and differentiate them towards a chondrogenic lineage. So in our earlier studies, we confirmed successful chondrogenesis using the iPSCs, and specifically we confirmed that derived iPSCs had superior differentiation ability towards the chondrogenic lineage. We confirmed the cartilage regeneration ability of the iPSC-derived chondrogenic pellets when we implanted them into a artificially made defect in the rat joint. And we also attempted to isolate the single chondrocytes from the chondrogenic pellets and inject them through intra-articular injections into the defect region. And so we confirmed both regeneration ability in the chondrogenic pellets and single chondrocytes, but we confirmed that the three-dimensional pellets showed more efficacy in regeneration. So through our previous studies, we confirmed that the three-dimensional chondrogenic pellets had more differentiation efficiency and therapeutic efficacy. And however, they have to be delivered through a surgical procedure, so they have less therapeutic accessibility compared to the single chondrocytes. So we tried to develop a size-reduced three-dimensional chondrogenic cell pellets to combine the benefits of both two-dimensional and three-dimensional platforms. And this was the first product called the minimal injectable unit chondrospheres, and we called it in short MUCON. To generate clinical grade MUCONs, we first established a clinical grade iPSC cell bank. And using the cells from the cell bank, we generated clinical grade MUCONs for further validation and preclinical studies for IND submission. So the generated MUCONs had a relatively smaller size, which was about 200 micrometers, and chondrogenic markers such as FOXP9 and collagen type 2 was highly increased in the differentiated pellets. Also, we confirmed increased secretion of ECM proteins, which is one of the main characteristics of chondrocytes. Through single-cell sequencing, we confirmed a distinct cell subpopulation in each step of differentiation, and in mature MUCONs, at fully differentiated MUCONs, a uniform cell type was observed as well. Also, genes related to cartilage development and ECM organization was increased in the MUCONs. So, through the characterization, we confirmed that the current products will successfully generate cartilage-like MUCONs in vitro. So after confirming characteristics, it was important for us to confirm the efficacy of the MUCONs. So first, we tried to confirm them in rabbit OA models by generating rabbit models through ACLT, which is anterior cruciate ligament transection. And after injecting them in MUCONs, we confirmed that the MUCON injected group had a more cartilage protective effect and had less destructive area. When we also implanted them in an artificially made defect, we confirmed that the implanted MUCONs were able to assemble with another and fill the defected areas. However, actually the rabbit model does not represent the human osteoarthritis, so we had to develop a OA model in a larger animal. So through our previous studies, we confirmed that the ACLT model showed less destructive cartilage characteristics, so it was hard for us to confirm significant efficacy by MUCON. And on the other hand, the osteochondral defect model was too artificial compared to the actual clinical features. So what we did is that we induced ACLT and also partially removed some part of the meniscus to induce a more OA-like defect in the canine models. Through this modeling, we were able to make a more OA-like model in the beagle dogs. And after injecting them with MUCON, we confirmed that a cartilage-like tissue covered the defected region. And through histological analysis, we confirmed that there were MUCON-like structures within the tissues covering the defect. We also confirmed the efficacy in many pig models. So we also induced ACLT and partially removed the meniscus. And additionally, we made them exercise regularly to further induce osteoarthritis. And in this trial, we performed gait analysis to evaluate the efficacy of MUCON. So, similar to the previous results, we confirmed that the destructive area in the cartilage was less formed in the MUCON injected group compared to the vehicle control. And when we observed this by MRI and X-ray imaging, we confirmed that the defected area in the vehicle control was covered by additional tissue in the MUCON injected group. ORCI score, which indicates OA grade, was significantly decreased in the MUCON injected groups. And when we compared the weight-bearing rate of the hind limbs, the MUCON injected groups had no significant difference throughout the weeks in each limb. However, the vehicle control showed consistent difference between the left and the right hind limb. But what's interesting is that we confirmed that a specific region showed more efficacy in the mini pig models. So we analyzed 4 different regions in the articular cartilage, and we confirmed that the dorsal part of the articular cartilage, including zone 1 and 2, showed higher efficacy than the other regions. So through efficacy testing, we confirmed that we were able to establish a proper osteoarthritis model for testing MUCON efficacy, and also that the intra-articular injections of MUCONs showed therapeutic efficacy in various animal models. And we also confirmed the safety of MUCON, and to check on the terminogenicity of the generated MUCONs, we first confirmed the negative expression of tron 60. And when we implanted them subcutaneously, we confirmed that there was no teratomer formed in vivo. We also confirmed the genetic stability of MUCONs and the original iPSCs through SDR analysis and somatic code invariant analysis and CNV analysis as well. So in summary, we developed a protocol to generate injectable chondrogenic pellets called MUCON. And in these MUCONs, we confirmed the cartilage-like characteristics. And when we delivered them by intra-articular injections in various animal models, we confirmed less cartilage destruction and improved symptoms of OA in the animals. Also, genetic stability was confirmed in generated MUCONs as well. For further studies, we are currently trying to establish a freezing protocol for the long-term storage of MUCON, and also we are trying to verify the engraftment of MUCONs in the larger animal models. Also, we're planning to do more efficacy testing in other animals such as goat or horses, and further to reveal the mode of action of the injected MUCONs. I am proud and also humbled to say that we recently got the final approval from the Korean FDA, and we are planning to inject 3 actual osteoarthritis patients with MUCON in the near future, and we are aiming to confirm safety and efficacy in human subjects. So I would like to personally thank my team at the Citizen Lab at Tesla University, and also the researchers at IPSO. And I would like to say that most of the animal studies was done in collaboration with K-Biohealth. Thank you, and I'm happy to take questions if I have any time.